Good morning. My name is José Jalife, and I'm chairing this session from bench to bedside in arrhythmias in mechanisms of sudden cardiac death. We have a wonderful group of speakers, a very international group of speakers from Europe through Spain, into the United States. And we're going to be talking about different aspects of cardiac arrhythmias in the young. And I'm really looking forward to this talk. And first, I would like to introduce our first speaker, Dr. Connie Bezzina from the Netherlands, who is going to be talking to us about monogenic and polygenic contributions to the risk of sudden cardiac death in the young. Connie, welcome. Thank you very much for joining us. I'm glad that you're here, at least virtually, and we're looking forward to your talk.
Thank you very much for the introduction and also for the great opportunity to present to you today. So indeed, I'll be talking about monogenic, and polygenic contributions to the risk of sudden cardiac death in the young. So essentially, in the last 2 to 3 decades, we've learned a lot about genetic predisposition for sudden cardiac arrest in young individuals. And classically, we've divided these disorders into the primary electrical disorders and the cardiomyopathies. And we've learned that in the primary electrical disorders, these are caused for a large degree by genetic defects in genes encoding ion channels. And on the other hand, those studies have taught us that the cardiomyopathies are caused by a variety of genes encoding structural proteins that are involved in cell-cell contact, etc. Importantly, those gene discoveries have taken place in the setting of the monogenic inheritance paradigm, meaning that there is an assumption that one large effect genetic defect, essentially variants that are very rare in the gen population, that carry a very large effect size, by themselves are enough for the individual to reach the threshold to display manifestations in the disorder. So essentially, those studies take place in pedigrees, extended pedigrees, such as this one shown in the cartoon over here. And one would study affected individuals, try to identify a chromosomal segment that is shared among all affected individuals. And once that chromosomal segment is identified, one zooms into that segment to identify the causal rare variant in the causal gene. Now, these discoveries have had a huge impact on patient care. And what we see here is a paper which came out recently, which is basically a paper which provides guidelines of how genetic testing can be applied in these disorders. And what we see over here in this table is the impact that genetic testing has on the prognostics, on the diagnosis, prognosis, and therapy in these patients. So where we see multiple pluses, that genetic testing is highly recommended, is indicated and is useful, etc. But so essentially we see a lot of disorders over here in the arrhythmia syndromes and the cardiomyopathies where we see a lot of pluses, meaning that genetic testing really has an impact in the management of patients with these disorders. Now, despite the fact that there has been a huge impact of genetic testing, there are still outstanding challenges that are faced in genetic testing of individuals with these diseases. One of them is reduced penetrance and variable disease expression. What does this mean? This means that within a family where the genetic defect has been identified, typically not all mutation carriers, such as this one shown over here in this cartoon, would display signs of the disease. And furthermore, among individuals that do display signs of the disease, we observe a large variability in disease severity. This is most nicely observed in the setting of founder mutations, so large kindreds, large families that share the causal genetic defect. And I like to show this example over here. This is the KCNQ1 A341V founder mutation that Peter Schwartz reported in a large kindred from South Africa. And what we see here, as expected, mutation carriers have prolonged QT intervals compared to non-carriers. That's expected. But what we also see among the mutation carriers is a very broad variation in the extent of QT prolongation. Furthermore, when we focus on symptomatology, we also see that while quite a few persons in carriers of this mutation display symptoms, and actually this is rather unusual because we know that this mutation is quite severe, not all individuals go on to develop symptoms. Another outstanding challenge that we face is that some patients actually remain genotype negative after extensive genetic testing of the known disease genes. One of these disorders is Brugada syndrome. We are able to identify a rare variant in SCN5A, be it missense or truncating, in about 21% of patients, yet we do not identify such a variant in the remaining 79%. And essentially, SCN5A at this moment remains the only gene unequivocally associated with the disorder. The same situation we have in hypertrophic cardiomyopathy, where genetic variants, where mutations in genes encoding for sarcomeric proteins prevail, yet we are able to identify such mutations in only half of patients. So another observation is that many times we're unable to identify the causal genetic defect in cases that present sporadically, or in any case, in situations where we have a less pronounced familial clustering. So a few years ago, while we know for sure that monogenic forms of these disorders do exist, we hypothesize that maybe not all patients actually have a monogenic or Mendelian inheritance. And we hypothesized that these disorders could actually be more complex, at least a subset of them could be more complex than we had assumed to date. So it could be that within a family there is a large effect, rare genetic defect, but that genetic defect by itself is not sufficient for the patient to demonstrate manifestations of disease. And essentially, the effect of the large effect genetic defect is importantly modulated by the co-inheritance of additional genetic variants, as well by other non-genetic factors, environmental factors. And in such a what we call a near monogenic or oligogenic inheritance paradigm, besides rare variants, one would need to accumulate different types of variants that are present actually in the germ population, be it common variants of small effect and intermediate variants of intermediate effect, that together with the rare genetic defect conspire to reach the threshold for the disease. A more extreme scenario is a polygenic inheritance, where one would need to accumulate multiple genetic variants to ultimately reach the threshold to display the disorder. So about a decade ago, we started investigating these more complex inheritance paradigms by starting to look at common genetic variants, asking the question, what is the role of common genetic variants in susceptibility for these disorders and severity among affected individuals? So the most commonly used methodology to identify susceptibility variants is the case-control genome-wide association study approach, where one compares large sets of patients to general population controls, and essentially one genotypes patients and controls on SNP arrays. Typically, these SNP arrays contain about 700,000 SNPs. We impute the untied SNP and non-genotyped SNPs by virtue of what we know about haplotypes and phasing in the germ population. And then we essentially test variant by variant as many as 7 million SNPs littered throughout the genome. And we ask the question, is there any variant that is significantly more prevalent in the patients as opposed to the controls? Because that becomes then a candidate susceptibility variant for the disorder and being studied. So till now, GWAS has been conducted in these 4 inherited cardiac conditions: Brugada syndrome, long QT syndrome, hypertrophic cardiomyopathy, and dilated cardiomyopathy. The papers are all listed over here. And today I will go through a few concepts of what we've learned, what these studies have taught us, and I'll bring examples from Brugada syndrome and hypertrophic cardiomyopathy to demonstrate what we've learned from these studies. So what did these studies teach us? First of all, these studies have taught us that common genetic variants, that is single nucleotide polymorphisms of small effect, play a role in all the inherited disorders that we've studied thus far. So here I show the latest study we conducted in Brugada syndrome, where, as an example, where we compared 2,800 cases against 10,000 general population controls. And this is a Manhattan plot that emerges from such a study. So every little point over here on this plot represents a single nucleotide polymorphism. As I said, this plot aggregates the data from 7 million single nucleotide polymorphisms that are tested. And what essentially the study showed us, that there are 12 different places in the genome that harbor common genetic variants that contribute to risk for Brugada syndrome in the setting of a polygenic inheritance. So what I forgot to say is that the single nucleotide polymorphisms are arrayed here per chromosome, and what we see here is the negative log of the p-value. So what we like to see are single nucleotide polymorphism that have a highly significant association, in any case below 5 × 10⁻⁸, because like that we're correcting for the total, for multiple testing of so many SNPs throughout the genome. This is another study that we conducted in hypertrophic cardiomyopathy. In this study, we compared 1,700 cases with HCM from the Netherlands, UK, and Canada, compared them to over 6,000 controls. And again, here, we identify 16 chromosomal regions that harbor common variants that contribute to risk of hypertrophic cardiomyopathy. So unequivocally, common variants contribute to risk likely across all inherited cardiac conditions. Furthermore, we've learned through SNP heritability estimates, actually we've learned that they not only contribute, but also they contribute quite appreciably. So essentially, SNP heritability estimates have been, that we found across the different disorders, range from 0.12 to 0.34. What does this mean? This means that between 12 to approximately 34% of variance in disease susceptibility is attributable to common genetic variation. And I think that's quite an appreciable attribution to variance and susceptibility. What we've also learned is that there can be different genetic architectures can exist for a given disorder. So we know, for instance, that long QT syndrome is primarily monogenic, but that can also coexist with also more complex forms. So this example actually that I show here is from hypertrophic cardiomyopathy. So what this study did, they calculated SNP heritability estimates now in HCM patients that are sarcomere positive, that is, they carry a rare genetic variant in a gene encoding a sarcomere protein, and they calculated it also in HCM patients that are sarcomere negative, so those 50% that have remained genotype negative. And what we see over here is that SNP heritability is almost double in the sarcomere negative as compared to the sarcomere positive. And this indicates that common genetic variation has a more prominent role, as we would expect in patients that are sarcomere negative. So essentially, within a given disorder, we can have a continuum of genetic complexities, where on one extreme, we have pure monogenic diseases, we can then have patients with a rare genetic variant where common genetic variants also contribute to the more extreme polygenic inheritance in sarcomere negative HCM. Also, we've learned that variants of different frequency and effect size can exist at a given locus. And a nice example of this is the SCN5A locus in Brugada. So we know that coding region variants are found in 20% of patients. There are loss of function mutations. They're either premature truncations or missense mutations that cause loss of sodium channel function. But what the Brugada syndrome GWAS showed us is that common genetic variation, so small effect, common genetic variation is also present at this locus. And what we've also seen is that these common genetic variants, they occur actually in the non-coding regions of the genome. They coincide with elements that we believe are important in regulating the amount of expression, that is the mRNA, the amount of mRNA of SCN5A that is made, and thereby also contribute to Brugada syndrome now, not through effects on the channel protein, but by regulating the amount of protein of sodium channel that is made. So essentially, these genetic variants occur at enhancer regions that in the three-dimensional structure of the genome make contact with the promoter of the SCN5A gene and thereby impact on the dosage of the sodium channel at the membrane. What we've also noticed is that a marked genetic correlation exists between genetic susceptibility to these disorders and genetic variants that actually contribute to endophenotypes that are involved in these disorders. So what are endophenotypes? By endophenotypes, we mean those traits that are measurable, such as ECG parameters, such as parameters of cardiac structure and function that we can measure by MRI. These all represent traits that capture the biology and biological pathways that we think are involved in these disorders. Now, as opposed to Brugada syndrome and all of these inherited cardiac conditions that are rather rare in the general population, we can measure ECG parameters and CMR in very large segments of the general population. So if we conduct those studies in hundreds of thousands of individuals, we can achieve a very high statistical power to identify those variants that are involved in these traits, which then represent very high priority variants that could form susceptibility variants for these disorders. So this is what we did actually in the study in our effort to identify susceptibility variants for hypertrophic cardiomyopathy. We conducted genome-wide association studies on 9 different left ventricular parameters of structure and function as measured by cardiac magnetic resonance. Once we conducted those GWASs, we could then test the genetic correlation with genetic susceptibility for hypertrophic cardiomyopathy. And you can appreciate that over here, we have a positive genetic correlation for some parameters as expected, for instance, for mean wall thickness, also for parameters that represent cardiac contractility. On the other hand, we also identified negative genetic correlation with left ventricular and systolic volume, for example. And what we did next, using sophisticated genome techniques, is incorporate now these GWASs together to enhance the identification of susceptibility variants for hypertrophic cardiomyopathy. So essentially, this was the Manhattan plot for the GWAS of HCM cases versus controls. This allowed us to identify 6 susceptibility loci, but when we incorporated the information of the genetic factors that contribute to these parameters in the general population, we were able to boost the statistical power of the HCM case control GWAS and double the number of associated loci with the disorders. What we've also learned is that disease susceptibility variants can also modulate disease severity. Of course, this is clinically very important in the management of families where a sarcomatic mutation, for instance, has been identified in HCM. So this is what we attempted to do here. Of course, I mean, common variants individually have a very small effect size, but if we start considering them in aggregate in the setting of a polygenic risk score, which essentially takes into consideration their aggregate effect, then maybe we can also start explaining differences in disease severity among carriers of the monogenic defect. So this we did in a set of 368 probands and their relatives with mutations in the sarcomatic gene. These were the 3 different genes that were affected in these individuals. Then in each individual, we calculated that individual's polygenic risk score, which essentially we sum up the number of risk variants that individual carries, and where each variant is weighted for the effect size that is identified in the GWAS, and then we relate the polygenic risk score among these individuals with disease severity. So we investigated a relation with maximal left ventricular wall thickness, which is of course a very important phenotype in hypertrophic cardiomyopathy, and could identify an effect of the polygenic risk score with maximal left ventricular wall thickness, and also we also could identify an effect on clinical events. I have to say that these are very early days. The effect is small. Because the sample size was very small, we had only 368 patients. We had to consider a composite endpoint of clinical events, various clinical events. But I think it is a proof of concept that as we identify more of these susceptibility variants, by incorporating them into larger polygenic risk scores, we might reach, hopefully, polygenic risk scores which could be used clinically. And lastly, GWAS loci allows us also importantly to generate hypotheses about molecular mechanisms of disease. And I here turn back to the genome-wide association study in Brugada syndrome. And basically what we did when we saw this Manhattan plot, of course, we want to use it also to learn about the biology, about the pathomechanisms involved in the disorder. So what do we see over here? I mean, as I said, we have common variants around the SCN5A gene. Nice, but not completely unexpected. We also identified a bunch of transcription factors, some of which have already been linked to regulation of ion channels in the heart. So actually we turned to genes that had not been in any way implicated actually in cardiac function. And one of these was the MAPRE2 gene. This was interesting because MAPRE2 encodes for end-binding protein 2, which is one of 3 end-binding proteins that are involved in microtubule biology. And essentially, earlier studies had already shown that EB1 is actually involved in the trafficking of connexin 43 to the cardiomyocyte membrane. So this allowed us to hypothesize, could it be that EB2 now is involved in the trafficking of sodium channel to the membrane? And in a very nice collaboration within our LEDUC network, we knocked out MAPRE2 in zebrafish and iPS-derived cardiomyocytes. And for the first time, we could demonstrate that MAPRE2 is involved in ventricular conduction and also is involved in sodium channel expression and therefore sodium current in cardiomyocytes. So where do we go from here? I think that we need larger GWAS and larger sets of patients for identification of additional modulatory variants. The identification of additional modulatory common variants will increase the discriminative capacity of polygenic risk scores in distinguishing between affected and unaffected individuals, but importantly also between mildly affected and severely affected patients, or those that will develop severe disease in the future. And the question, I think, is whether we will be able to identify sufficient SNPs to allow us to reach these discriminative polygenic risk scores that will be sufficiently discriminative for clinical utility. Also, all these studies that I show you have been primarily conducted in patients of European ancestry. We need work in patients of non-European ancestry. Also, till now, we've studied the common variants of small effect, but for sure, intermediate effect variants with an intermediate frequency in the GAN population also play a role. For this, we need to start moving into studying exome and genome sequencing data for the identification of those variants. And also we have a need for more work in the functional domain, because essentially locus discovery has now far outpaced the gene validation studies and studies that aim to provide mechanistic insights. I would like to acknowledge all the collaborators that have collaborated with us in the HCM study and in the Brugada study. Of course, these are huge studies that require many patients, and we're really indebted to all the clinicians that collaborate by providing patients to include in these studies, and also our collaborators in the functional studies within the LEDUC network. And I would like to thank you for your attention. Thank you.
Thank you very much, Connie. Unless there is a very, very important question, we're going to leave the discussion to the end. So the next speaker, I'm very happy to introduce, is Dr. Mario Delmar from New York University, who is going to be talking to us about molecular substrates of sudden cardiac death in young athletes, desmosomal reserve, and the good enough solution. Mario?
Thank you very much, Pepe, and thank you all for attending this meeting. It is really an honor to be here. It is fantastic to receive an invitation to come to this beautiful city, to this great meeting, and to see good old friends and make new ones. I will first acknowledge the people that have worked on the project that I will present to you today. These are those in my lab. There are many others, but these were the primary ones that participated in the study. And also collaborations that I have. Do I have a pointer? No. In where? Is there a pointer?
We need a pointer.
And
you use the computer?
It doesn't come up. Oh, there it is. I have it now. I have it on the end. Oh, that one. Anyway, people also in other centers, particularly colleagues in Utrecht University in the Netherlands and at Hopkins. So my lab is interested in cases like this. This is the heart of a 16-year-old boy. He was seemingly healthy. He was playing soccer with his friends, football, if I am in the right country to call it the right way. And all of a sudden, this kid, as he's running, loses consciousness, falls, and efforts to resuscitate him are unsuccessful. To the left, you see the postmortem MRI. And what you can see in this four-chamber view is the total loss of muscle mass in the right ventricular free wall that on histological section has been replaced by fat, which comes out in white, and with collagen, with fibrosis, as it comes out in blue in this staining. The muscle stains red, and as you can see in the section of the right ventricle, which is this one, there is a major loss of the muscle mass that has been replaced by fibrofatty tissue. On the left, you can already see some collagen infiltrates, but the left ventricular mass is still good enough to maintain contractility, and therefore this kid is completely asymptomatic until he unfortunately falls. This kid is diagnosed with arrhythmogenic right ventricular cardiomyopathy. This is an inherited heart muscle disease that may result in arrhythmia, heart failure, and sudden cardiac death. But importantly, these are not consequent to each other, or even they do not develop in parallel. We have shown through the years that the arrhythmogenic component of what I will refer to as ARVC and the cardiomyopathic component have different molecular substrates and can happen at different times. And in fact, in this particular disease, the arrhythmogenic component is most often the first symptom of the disease, and in fact, sudden cardiac arrest can be the first manifestation of the disease in about 50% of the patients in a young age. ARVC has been linked to mutations in desmosomal proteins. For those of you to remember your years ago cell biology, the desmosomes are essentially molecules or complexes that form Velcro between cells. They are there to hold the cells together so that they can mechanically work together. An estimated 50% of the cases of the familial variant of the disease are linked to mutations in the gene coding for one of these desmosomal proteins, and is called plakophilin 2. I will refer to it as PKP2. Now, that is about 50% of the cases of the familial kind. What happens to the others? What are the molecular causes of the disease in other patients that do not have an obvious primary dominant mutation? So it's already 10 years ago that André Legerche brought up this concept that the right heart in athletes presents a similarity to the right heart of patients with arrhythmogenic right ventricular cardiomyopathy. And this has been reviewed through the years. This is one review done by Brugada and his colleagues, where they actually compared the phenotype of ARVC and the phenotype of the athlete's heart. And there are similarities. There are also differences. And most recently, actually, we put together this review with our colleagues at Hopkins and in Zurich, where we present the hypothesis that the molecular pathways that lead to the disease, they may represent in the athlete heart a mimicry of the molecular pathways that go to ARVC. So we actually stem from this study that I think is one of the most important studies in the ARVC field, done by Cindy James and Hugh Calkins at Hopkins, where they actually did a very careful analysis of the amount of exercise that their patients had done, and developed a survival plot, basically a Kaplan-Meier plot, where they separate those that are non-athletes in blue and those that are athletes in green. And they look at the survival from the first sustained VT-VF. And you can see that in the athletes, the curve drops much faster than in the non-athletes. When you do the same thing, but for heart failure, you can see that whereas the non-athletes actually have a very good heart failure-free life through the years, those that are athletes actually suffer from the heart failure at a much earlier age. So this showed that for ARVC in particular, exercise is actually a risk factor that actually accelerates the progression of the disease. So we decided to look more carefully at this by using experimental models and see what are the differences and what are the similarities that one can find between these two cases. And this is the paper that I will describe, in which we compared exercise versus deficiency of plakophilin 2. So this paper was published in a European Heart Journal a few months ago, and this is basically the model that we used. We are studying mice, and of course, men and mice are different, and there are many similarities amongst us, but the differences are obvious. So we took mice that are controlled, they are wild-type mice, and we put them on a treadmill. We put them to run 1 hour a day, 6 days a week for 6 weeks. And we are going to compare what happens to the overall transcriptome of these mice to what happens to mice that are sedentary, but that have a loss of the gene coding for plakophilin 2, for PKP2. So are these trained mice showing the changes that correspond to the normal physiological remodeling from exercise? The answer is yes. You can see that their left ventricular ejection fraction goes up with time, and they are running for 6 weeks. So after 6 weeks, they have an improved left ventricular ejection fraction, and they have an increase in the left ventricular anterior wall during systole, and also during, I'm sorry, this is shifting, but also the posterior wall. So, okay, so they are exercised. They are showing the remodeling that you would expect in the mice, or in the, sorry, in the athletes after that period of training. So, as you know, one nowadays can take a heart or any other tissue, and you can look at the entire family of RNAs that are inside that heart. 45,000 transcripts, 45,000 different transcripts. And if you take the transcript from a mouse that has been exercising, and you take it from the mice that have been sedentary, otherwise treated equally, you can then take the abundance of each single one of those transcripts and compare them and determine whether there is more of or less of a particular one. Each point in this plot of 45,000, each point is one gene. And what you are presenting here is the magnitude of the difference against, in the y-axis, the significance of that difference. Those that are to the left of us are downregulated transcripts. That is, there is less of them. And to the right, there is more of them. And what I want to bring your attention to is the genes that are marked in red on this left side, the desmoglian 2, desmocolin 2, plakophilin 2, ankyrin B. These are desmosomal genes. These are not mice deficient in a desmosomal protein. These are mice that ran, period. And what we find is that key molecules that are involved in maintaining the integrity of the intercalated disc are downregulated by exercise. In fact, one molecule that has been studied in the GWAS abundantly for survival, from sudden death is NOS1AP, and NOS1AP is also a molecule that shows to be downregulated by exercise alone. Now, we had done previously the same exercise, the same experiment for mice that were deficient in plakophilin 2, and when we looked at pathways that are downregulated in the exercised mice that are otherwise normal and tried to find out how do you put all of these differential molecules in one package, the package that is most significantly affected is the one at the bottom, arrhythmogenic right ventricular cardiomyopathy. So when we looked in a non-biased way at the transcriptome of mice that have been exercised, the most common pathway that results is ARVC. So we took the transcriptome that I just presented, and we took the transcriptome that we obtained from plakophilin 2 deficient mice that were sedentary. In this plot, I am plotting the magnitude of the change in the mice that exercised against the magnitude of the change in the mice that lost their plakophilin 2 expression. Each dot represents each single gene. And what you can see is that the first quadrant and the third quadrant of this Cartesian plot are heavily populated. In fact, in your mind, you can pretty much trace a good straight line between the two, meaning that when you compare the transcriptome, the changes in gene expression that occur resulting from exercise, and the changes in gene expression that result from losing plakophilin 2 expression in a sedentary mice, they correlate very well. In fact, if you take now plakophilin 2 deficient mice and you exercise them, that is really bad for them. In the sedentary mice, which are the purplish color, with 3 weeks of training, with 3 weeks after knockout of plakophilin 2, they lose about 10–15% of ejection fraction. But if you make those mice run, they actually don't complete the 6 weeks. After 3 weeks, their ejection fraction is below 40%, and they are unable to run. So yes, in these mice, exercise is also bad for them if they don't have plakophilin 2, because there is an additive effect of the two components. So what does this mean? And I really take from the presentation that Connie just did, beautiful work. If you just create this imaginary model of what is happening. To the left, we have we have cases in which we are looking at the abundance of molecules of the desmosome. How much of them are there? And there is a red line at which if you cross it, if you go under that line, now you are going to have a disease. Now, if you are a control person that live a happy sedentary life, you are way above the line for the threshold for disease. And if you exercise, you are losing some of that abundance of desmosomal molecules, but you are still above the red line, and there are other obvious beneficial effects of exercising that are maintaining you in actually a better cardiac health. Now, if you start on the right side, not necessarily with having lost plakophilin 2, which is a big, major change, but if there is a combination of factors, a multivariant effect that already reduces your desmosomal abundance, now you are closer to the threshold for disease. You have lost some of what we call a desmosomal reserve, to the point that now if you exercise, you easily cross that threshold, and now you're going to develop the molecular pathogenesis of ARVC, loss of apoptosis, cell death, and then the symptomatic case. A very nice way of thinking about this comes from these early studies in which they described what they called the good enough solution. The good enough solution can be very much simplified into a basic concept. The color of the eyes. Green eyes are good enough solution for seeing. Blue eyes are good enough solution. Brown eyes are a perfectly good solution. No eyes, not a good enough solution. In the same way, you can have different molecular composition that allows you to be okay. They are good enough until you break it into a different pattern. This was actually formally described, not with the example of the color of the eyes, but in this study in which colleagues took a very, very simple circuit in the lobster, in the neuron in the ganglia of the lobster. And after eating the tail, they looked at the different currents that compose the action potential in these lobsters. There are 3 simple currents. Each dot in there is one lobster. And you can see that the amplitude of the conductances for these 3 currents are different. Not every lobster has the same amount of current. If they look at the amount of copies of mRNA, each lobster had a different amount of copies for each one of these currents. But when you look at the action potentials, which are at the top, they all had the same action potential. They all solved it differently, but they came up with the same solution. This was exemplified in this work by Jim Weiss and his group some years ago. And actually, it kind of makes sense. I don't think that right now, I'm going to assume that we all have more or less normal QT interval, but I don't think that each one of us right now is solving our QT with exactly the same number of copies of the exact same molecules. We are all solving it differently. But in this case that Jim did here, you have 2 individuals, the blue one and the red one. And the arrow is pointing at the amount of IKR that each one of these 2 individuals has. The one at the top uses a lot of IKR to repolarize its action potential. The one at the bottom doesn't, uses pretty much not. Both of them have the same action potential. But now, if you give these 2 individuals a drug that will block IKR, the one at the top that depends a lot on IKR to repolarize its action potential is going to have an arrhythmia. The one at the bottom that doesn't is not affected by the drug. Each one of them was perfectly healthy until something came out from outside that made them reveal the arrhythmia. And what is that is obviously a major question that has a lot to do with what Connie just presented. So this is the model that I leave you with. It's a condition in which we are all solving our action potentials. We are all solving our intercellular junction. We are all maintaining the structure of the heart by a different combination of molecules in which we all have an enormous different collection of SNPs. Now, in most of our cases, we can tolerate exogenous stimuli, because even if we lose the representation of a particular set of molecules, we are able to maintain normal health. However, if you press the system, because you have the different combination of molecules, the different combination of SNPs, the nature of which for ARVC we do not know. We look forward to some study coming on the ARVC field of GWAS, like the ones that have been done for other diseases. Then we are at risk of developing the disease, even if it is an individual that when he or she is genetically tested, shows that there are no mutations in the genes coding for the desmosomal molecules. This individual may actually have the combination of SNPs that brought them back down below the red line and into the disease field. And so there is probably a molecular continuum between the athlete's heart and the ARVC. Now, I leave you with this line, which I love: all models are wrong, but some are useful. So what I have presented to you is in mice, and we are looking at transcripts. And yes, we have a totally different molecular configuration, but we use the models as a way of establishing particular molecular characteristics that allow us to examine hypotheses that cannot otherwise be tested in the patients, but that can lead us to practical hypotheses that can be advanced in the treatment of the general population. And with that, I thank you very much for your attention and for this invitation.
Thank you, Mario. I'm proud of you, but sit down. We're going to go to the last presentation by Dr. Juan Jiménez Jaimez, who is going to talk to us about short QT syndrome, a rare cause of sudden cardiac death caused by organelles and plasma membrane ion channel mutations. Juan?
Thank you. Well, thank you very much to the organization, Pepe, for this talk. This is a very nice and stimulating talk because most of the topics I'm going to treat now, we don't know. So it's very, very stimulating. I have no conflict of interest. This is a picture of short QT syndrome now, in 2022. Okay? So the frustrating thing is that only 20% of the cases with the available tools, we are able now to diagnose, to establish a definitive genetic cause. This means that 8 out of 10 patients that we attend in our clinic, we finally don't know which is the gene, the disease-causing gene. This is frustrating and at the same time stimulating. And besides, we can even appreciate in the families that have been published in Short QT that they are quite small. When we dedicate to the familial inherited cardiac diseases, we know that it's very important to have large families to extract robust conclusions. And these are the first 6 genes that have been linked to the short QT syndrome, and only 3 of them have an almost definitive causality that can be defended. The others, the remaining, they still have many doubts because they are de novo mutations and, at some point, doubtful experimental conclusions. So we feel the same when we see short QT syndrome as we, the physics and the astronomers must feel when they look at the universe. They don't know to explain any of these very far stars, and you must feel frustrated but stimulated to investigate. And that's what we do with the short QT. This is a very, very recent paper in the European Heart Journal that correlates or analyzes the validity of the pathogenicity of these now are 9 genes that have been linked to short QT syndrome. As you can see here, only these 4, the 3 first genes that were linked to the short QT, and another one that I'm going to comment in a couple of minutes, have a moderate to robust, strong association with short QT syndrome. This has been evaluated by an independent cohort of geneticists that have established the phenotype-genotype segregation, the validity and usefulness of the experimental studies. And as you can see here, some of the calcium, most of them calcium and sodium channels, have a very, very weak relationship that can be defended with the short QT syndrome. So even though now we only have the 20%, some of them generate a lot of doubt. So are we looking onto the right direction? Are we searching in the correct site? So this is the typical clinical scenario in short QT syndrome, according to what I have said, that we don't know the genetic basis. So we have to research, we have to look for, to try to give a reasonable explanation to this clinical case. This is a 30, now he's almost 20. When we started with him, 30-year-old male, asymptomatic, referred for familial sudden cardiac death. We had no autopsy from the father, but yes, from the brother. You know what is a blank autopsy. We did not see absolutely anything. That doesn't mean there is no anything. Of course, he has suddenly died, so something must have happened. And when we perform the basal ECG to the proband, we see a very extremely short QT syndrome. It's likely that sudden death in the two relatives is caused by a short QT syndrome, and we try to find a reasonable explanation, but the NGS panel, with more than 200 genes, including, of course, this I have commented before, were completely negative. So this is the typical case, 8 out of 10 in short QT cases. So we have to rely not only in genetics, but also in clinics, but also, of course, in the central figure of the role, the diagnostic role of the ECG, which is the hallmark of this disease, the short QT syndrome. And, of course, finally, the familial assessment, which is not completely or clearly the same as genetics. Genetics can be negative, but the disease keeps being inherited and familial. Looking into the clinical data that we have for short QT, of course, we have not very, very large series. We think this is an extremely rare condition. We don't know if we are seeing just the tip of the iceberg. Perhaps we are seeing only the most serious cases, and the asymptomatic ones are hidden inside the population. But this is what we have. This is the largest series published up to now, and we see that the only strong predictor of sudden cardiac death is having suffered a sudden cardiac arrest, okay? Because asymptomatic patients, sometimes they have an event, but they clearly show a different clinical outcome to symptomatic with cardiac arrest. Syncope is very uncommon, can you see? And what is most important of this graphic is that the length of the QT interval, which is in the opposite disease, the long QT, is clearly related the length of the QT interval with the incidence of cardiac events. Here it's not so clear. Perhaps it's a matter of sample size, but you can see that cardiac arrest even is more prevalent in longer QTs than in shorter ones, where we can find even patients that are asymptomatic. Another clinical picture of the disease: males are suffering the disease with a higher incidence and a higher incidence of adverse events, malignant arrhythmic events, and it seems that hydroxyquinidine seems to be effective to prevent serious malignant arrhythmic events. Okay, when going into the diagnostic tools, ECG, basal ECG, we do not have an unmasking tool as we have for the long QT, for the catecholamine VT. We must rely only on the ECG, and there is a problem. The problem of the penetrance, the problem of the different expressivity on the ECG. We can suffer short QT with 30 and 70 milliseconds, and we can suffer it from a variety of measurements of the short QT. So we have a diagnostic criteria that have been updated sometimes. Currently, we can clearly and definitively diagnose short QT when we have a QT corrected interval below 340 milliseconds. And if we have 360, we need to add a different, another condition in the same patient that is related either to family history of pathogenic mutation of short QT or to the presence of an idiopathic, previously named idiopathic ventricular fibrillation in this patient with a borderline QT-corrected interval. But do we know, this is a very, very known slide, do we know how to measure QT? Of course, it's not only for general practitioners, internal medicine, cardiologists, even electrophysiologists fail to correctly measure the QT-corrected interval. So you see, this is the hallmark of the disease, the first step to diagnose your QT, and we even don't know how to measure the QT-corrected interval. Some additional markers that are not included in the diagnostic criteria, the TPIC-TN, we will see later. And finally, genetics. genetics and familial assessment. This is our family. This is the family of the boy I presented before. This is one is the boy. We have, as I told you, a negative NGS panel. And what else can we do? This doesn't mean that we have to do cascade screening. We perform a cascade screening assuming that they were short QT, and the surprise was that we have in the asymptomatic cohort of patients 5 more carriers of, not carriers, phenotype positive patients for the short QT, which are marked here in black, which is the difficulty when facing a family with sudden cardiac death. Sometimes we don't have available data. This woman lived until the 90s, completely asymptomatic. She only developed, and we have an ECG that I will show you later, atrial fibrillation, but no syncope, no cardiac arrest. This is intriguing when we try to establish an outcome prediction. And another problem is that they usually have some familial problems that are not clearly the mutation. For example, this one, who is not positive, doesn't speak to this and to this. All of this live out of our city, and it's quite difficult to try to compile all the clinical and genetical data. But even though we try to rescue the ECGs from the patients that have suddenly died before. You see, this is the ECG of the father that had suddenly died before, and we had no autopsy, okay? Extremely short QT and a very abnormal QT and T-shape, the T morphology. I don't know if you have previously seen this kind of T-segment elevation, very strange, quite strange repolarization with extremely short QT. This is something that was present not only in the father, but also in one of the sisters that is currently alive and lives very happy and asymptomatic with 60-something-year-old. This is a representation of the ECGs of all patients that we have diagnosed with the short QT syndrome. And what about genetics? Because genetics were negative, and the title of my talk today is a disease of the ion channels from the cell membrane and from the organelles inside the cell. What's that? Well, that means that if you are unable to diagnose the genetic cause of a short QT when looking on the cell membrane channels, perhaps we have to look, because with that strategy we only achieve the diagnosis in 20%, as you see here. Perhaps we have to look inside the cell. The first time I talked or I said this to a professor, it was not Dr. Jalife, another one, he laughed at me a lot and said it was ridiculous. No, the problem must be in the membrane. And I said that because we have done previous experiments that I will show you now, and because at some point there have been some papers describing intracellular proteins related to shortening of the QT interval and malignant ventricular arrhythmia, such as this one, which is a mutation of a transporter inside the cell, which is developing a dilated cardiomyopathy with extremely short QT. This is a case from our cohort that we cured, treating it with carnitine, lengthening the QT interval and going up to normal range, the ejection fraction. This has led to a very, very recent communication in the last European Congress, where it's postulated that perhaps L-carnitine could be a treatment to prolong the QT interval for these patients and prevent ventricular arrhythmias. And this is the second paper about intracellular proteins that can cause short QT and sudden cardiac death. How? Well, this is a very, nobody would have thought to look inside this protein when facing short QT, but now we have these tools, the large NGS whole exome sequencing panels and GWAS that have been said before, and they found this mutation in this very, very strange exchanger that is inside the cell that when translating into a murine model, they demonstrated that the knockdown murine have a alkalinization of the cytoplasm that would lead to control the membrane potassium channel to leave them open. So demonstrated a short QT. And this is a very large family. This is what we like. When we do research on inherited cardiac conditions, we want very large families to try to establish a good correlation between the genotype and the phenotype and try to demonstrate that this is a causal mutation. And this is one of the genes, of the 4 genes that I told before in the table, that has a consistent relationship to the short QT, and it's inside the cell. So what did we do with our family? We took blood from 3 carriers. Well, carriers, no, phenotype positive patient with short QT, we took their blood, and from a negative phenotype patients, and we put them inside the machine, the whole exome sequencing, okay? We did an exome of the fourth and tried to see if they have any variant in common, the 3 carriers, and that was not present in the non-carrier. And we found this very, very strange lysosomal potassium channel. This is an intracellular organelle, as you know, the lysosome, and the current relationship between the lysosome and the cell membrane is yet to be elucidated. We don't know, but we are trying to demonstrate a relationship. What is clear is that there is a good correlation between the phenotype. All non-carriers have normal QT intervals, and all carriers, we were 7, including obligate carriers that are here. Okay, this father, we don't have DNA on him, but we assume he's a carrier. And this lady that died a long time ago, we don't have DNA, and of course from the boy, but we assume they must be carriers, okay? So there is a good correlation of this mutation that is completely absent in control. How can a lysosome mutation affect the action potential? We have some hypotheses, and we try to demonstrate this, as I'll tell you in a minute, contacting with Dr. Pepe Jalife, as I will tell you later. Which are the 2 hypotheses? Direct incidence to the potassium channels in the cell membrane, or indirect here, by not degrading the potassium channels that may be open when they should be destroyed. Something like that. Okay? First of all, we contacted with the group in Pennsylvania who discovered this very strange mutation, and they did. This was frustrating when they said that this is a loss of function mutation, because we expected a gain of function. If it's in the cell membrane and it's causing short QT, we expected a gain of function, but no, it's a loss of function mutation. So we decided to contact Dr. Jalife and try to create a murine model, but that still is going on with experiments. I'll show you some pictures, but now we are purifying the model and trying to finally recapitulate and demonstrate the phenotype in carriers of this mutation. These are experiments from Dr. Jalife's lab, and I will not go deep in them, but they showed us that at the very, very first experiments, that needs to be confirmed, that the mice carriers of this mutation showed a shorter QT than controls and a shorter action potential duration than controls. So this is very stimulating. What is remaining is try to elucidate. They even showed spontaneous arrhythmias. Dr. Jalife can explain to us in a minute, spontaneous arrhythmia during this phase 2 of the action potential. And, well, I won't show these 2 experiments because truly I don't understand them very well. I tried for Dr. Jalife to try to explain to us if we have any hypothesis to link this lysosomal potassium channel to the shortening QT. So finally, I would conclude saying that this is a very extremely rare condition. We don't know if perhaps there are a lot of asymptomatic patients hidden with a suspected high incidence of adverse arrhythmic events. Familial assessment is essential, and the ECG plays a central role in diagnosis. New genetical targets are needed to identify causal genes. These genes might not be or might not codify for cell membrane proteins, but intracellular proteins. And thanks for your attention.
Thank you very much, Juan. So now I have all of the speakers with us, and I will open this session for discussion. And I will start by first thanking Connie for being here with us and for giving us a wonderful talk. It was a great summary of the work that you've been doing, the wonderful work that you've been doing with both Brugada and HCM. A question I have for you, Connie, is what is the role you showed us some examples of huge families, the one that Peter Schwartz studied in South Africa, in which there was a lot of the variability in the penetrance as well as in the symptoms. So what is the role in that, not necessarily penetrance, but in the symptoms of post-translational modifications that are occurring not only in the sodium channel or whatever the channel is responsible for this syndrome, but other partner proteins that might be modified by the mutation itself. Can you tell
Yes,
us about
sure.
it?
I think that's a very good question. And actually, I think that is what actually the study in Brugada, are you still there? Because
Yes.
okay,
Yes.
good.
Yeah,
I don't see you anymore.
You are loud and clear, so
Yeah,
please go
so
ahead.
basically, indeed, I mean, those are very probable mechanisms, right? I mean, pathways that funnel into the function of the main channel that is leading to the disorder are high priority pathways. And genetic variation that impacts on processes that are involved in post-translational modifications of channels are highly probable. And actually, this is, I think, what the MAP-RE2 study showed us. So we knew, that coding region variants in SCN5A impact the channel function. Non-coding region variants in SCN5A impact the channel dosage. And now the MAP-RE2 functional analysis showed us that impacting on pathways that are involved in channel processing, be it trafficking or be it, glycosylation, will also impact the disorder. So absolutely, I think that is highly likely happening. And I think the MAP-RE2 work provides evidence for that directly, if that answers your question.
It does, but how do you study those in the context of the GWAS studies and in these patients?
Yes. So essentially, in long QT syndrome specifically, we've conducted a GWAS. We have identified also common genetic variants that impact on risk. In that study, we only took probands, and we're actually quite disappointed that when we looked for a relation between a polygenic risk score based on QT SNPs and disease severity, we couldn't find a relation. Now, our hypothesis there is that we took probands only. So we're sitting at, in the corner of the severe patients. So what we're doing now, we're expanding that study. We're taking again those probands, but now including all their relatives that carry the familial mutation, not selecting on phenotype. So we take, unaffected, less severely affected patients, and now we're gonna test that hypothesis again in hopes that we will find an impact on disease severity. And an indication for that is when we took SNPs that impact on the QT interval in the general population, we found a correlation, a high correlation between the effect they have on QT in the general population and their odds ratio for susceptibility to Brugada, so to long QT. So yeah, I think we just need to do more work.
Thank you. Anybody has a question for the panel? I cannot see you, but somebody's pointing at you.
I have
Can
a question
identify yourself, please?
Yeah, I'm Javier Ramos from Hospital Clínico in Zaragoza, and well, I learned from Dr. Jiménez Jaimez, and we're investigating, trying to elucidate some concept of arrhythmogenic cardiomyopathy and exercise. So I have a few questions for Dr. Delmar, and
two. Okay.
The first one is, what's your opinion about the exercise amount? I mean, when you experiment in the mice, do you have different exercise intensities? And also, in this mouse, is there any, I mean, not just triggering for a phenotype, development of the arrhythmogenic phenotype, but also as a trigger for sudden cardiac death? What's your opinion about that? And also, what about non-classic desmosomal ARVC, but ALVC or biventricular arrhythmogenic?
Yeah, thanks. Remind me of the first question.
The first one was about the intensity of the exercise. I mean,
so the intensity of the exercise actually has been better studied in the humans than in the mice. You have to understand that in the mice, it's sometimes hard to convince them to run. Also, it is hard to convince the students to do this running at night, which is the mice are nocturnal. So these are put on a treadmill, they start running, and we start each run with 12 mice. We have a treadmill of 6 lanes. We do 2 sets at the beginning. So, and then naturally, some mice actually like to run and some don't. And so the ones that don't would rather commit suicide than continuing running. Changing the parameters is very difficult. At the end of at the end of the whole 6 weeks, you end up with 4 mice that are going to give you data. So we decided to just stick to one same protocol. That being said, of course, it is possible. But in the studies in humans, actually, there is a much better correlation. And at present, if you ask Hugh Calkins, which is probably one of the clinicians in the US with the most experience in ARVC, he'll tell you, look, if you have a kid that you think is at high risk of developing ARVC, you may want to start talking to the family about orienting that kid towards the chess club rather than to the football team. And patients that have already developed the disease, he says, you know, golf, light walks, those are physical activities that are okay. Anything more than that, it is better that you don't guide them in that direction. Now, there are opinions about it, but then in the case of the triggers for the arrhythmia, we have studied that extensively in the mouse model. And we believe that there is a plakophilin-related dysregulation of intracellular calcium. And DADs are a major trigger of arrhythmias in these patients. So there is a structural substrate, but there is also dysregulation of calcium as a trigger. And we are working, we have an NIH-supported clinical trial to study flecainide as an RYR2 blocker in patients with ARVC that have an ICD in case it goes the wrong way with the flecainide. And the results of that should be coming out next year, probably.
And what about the non-classic desmosomal ARVC?
The non-classic desmosomal ARVC, I mean, it's, I guess, a question that I would put also towards Connie, right? At some point, you have to decide what's sarcomeric and what is not sarcomeric. Maybe the GWAS has identified new sarcomeric molecules that we thought they weren't. In the same case, I mean, yes, there are non-desmosomal molecules, but not every, I tend to say this, we classified molecules by the very first thing we discovered them to do. So connexin, we classify it as a gap junction molecule, but that's not the only thing it does. So in the same way, plakophilin-2 is a desmosomal molecule, but turns out to be a great transcriptional regulator, all the way into the nucleus. Yes, the non-desmosomals are because on the way between the intercalated disc and the nucleus, plakophilin-2 has a lot of a chain of events that lead to the dysregulation of transcription. And you can come into that pathway through many ways. And even though particularly us basic scientists like to draw arrows that go between one event and the next, there are many arrows that are coming in and out of that same path. And so I think there is a common path, and we can talk more, I don't want to belate this discussion, but there is a common path towards ARVC, but you can come into that path either starting at the intercalated disc, or through the mitochondria, or through calcium regulation, or through nuclear envelope, or through dysregulation of transcription.
Thank you very much.
Other questions?
I'd like to comment on that, because congratulations for your works. They are very interesting. The problem I see with the exercise in ARVC is the threshold of the exercise. We have now done an experiment with measuring the exercise with an accelerometer in a cohort of 70 patients with arrhythmogenic cardiomyopathy, desmosomal and non-desmosomal. And we have seen that dramatically 70% of patients with ARVC are sedentary, totally sedentary. So this could involve the balance would go to have events in another way, like diabetes, cholesterol, and finally cardiovascular events, not arrhythmic related, but they are as bad as arrhythmic. And when I see the 6-day running of the mice and how in the previous literature the exercise was measured, I see a problem with the threshold. What can we say to our patients? Don't do anything? That's not correct. I think this exercise is quite vigorous, and we all agree that athletes and competitive sport is leading to more advanced phenotype. And what about the moderate or the mild exercise?
Yeah, I mean, I think we all saw during the European Cup last year this player from Denmark falling on the field. This is a highly trained athlete, and other cases like that are present. I think that there is one of our colleagues in NYU that says, look, you get a 14-year-old that is very intense into the football team, and you know, are you going to tell him your teenage life as you know it is over because you cannot play anymore? Or, given that he does not have symptomatology but he's at risk, we let him keep playing, because when he gets to college and finds that there are other fun things to do other than being in the soccer pit, he's going to stop playing anyway. So, you know, these are the decisions that you guys face constantly. I think that in the experience of the study of the Hopkins group, there is evidence, and there is also experience coming from Norway, and from Utrecht, there is evidence that exercise is a risk factor. But how do you modulate that with your patient when you're facing this particular individual? Obviously is a tailored thing that you cannot just define on a universal statement.
Okay. Any more questions from the audience? Well, I'm going to go back to Connie again and ask her, I mean, nowadays epigenetic modifications have become fashionable, and genetics and epigenetics are mother and sister. What can you tell us about the epigenetics? How do they play in this grand picture of ion channel diseases and cardiomyopathies?
Yeah, I think it's actually a black box in our disorders, to be honest. That is a bit my impression. I think we haven't started looking into that, but I fully agree. It's something we need to do. And one reason why we haven't started looking into that has also to do with costs, because these arrays are still very expensive. But I think it would be a really great experiment to do. And I think it's also a point of intersection with environmental factors. Which I think will be very, very interesting. Absolutely. Yeah. Absolutely. Yeah, I think we need to start thinking about that.
Thank you, Connie. Any more questions from the audience? Mario, you have a comment?
Just one very quick comment. In this same paper, we worked with the Hopkins and Utrecht groups to look at the MRIs of the patients that were athletes. And when they came, they actually took the MRI and they diagnosed them, versus MRIs from people with the same training, but that did not have ARVC. And actually, wall thickness was different between the two. So there was an early correlate there. On the first diagnosis on the athletes with ARVC do not properly compensate, do not properly activate compensatory hypertrophic programs that other athletes should. So that it all goes to the moderate exercise that wouldn't go into compensatory remodeling, maybe off the hook. But the one that does an exercise that requires compensatory remodeling, that's the one that cannot properly compensate transcriptionally and then falls into the disease state.
What's your explanation for this exercise, the damage of the exercise to the desmosome? Is just a question of tension, or?
I actually think that it is tension. So we have a paper hopefully coming out soon in Circulation, where we looked at the relation between plakophilin 2 and the nuclear envelope integrity. And actually, in mice, in very early stage, you see that the nuclear envelope starts to lose its structure. So and the nuclear envelope integrity depends on the cytoskeleton holding it together. So we think that there is mechanical dysregulation at the intercalated disc that creates a chain of a domino effect on the integrity of the nuclear envelope. The nuclear envelope starts to lose its integrity, becomes leaky for calcium, and that is the beginning of DNA damage. And DNA damage leads to change in transcriptional program.
Yeah, because it's curious that we don't see this kind of close relationship between exercise in any other disease. hypertrophic, dilated, even ischemic. Exercise is very good for ischemic patients.
Exactly.
So it's surprising.
Because the cytoskeleton remains intact. But in here, the cytoskeleton does not remain intact. And one, you were talking about non-desmosomal mutations, one convergence, all differences accepted, are cases of lamin A mutations that simil ARVC. And that may be because the TMEM43, of course, is a nuclear envelope protein. So it may be the point of convergence is at the nuclear envelope. And indeed, you need a good intercalated disc to hold a good nuclear envelope integrity.
Interesting.
Now, I want to bring another question that might be far-fetched, but I think it's interesting. Yesterday, Stefan Hatem gave us a wonderful talk about the role of epicardial infiltration of fat and fibrofatty infiltration in atrial fibrillation. Is there a common theme here that we're seeing? How do you look at it?
Yeah, so we put forth a hypothesis years ago that the epicardium, which if you look at the EM, the epicardium, as you probably all the audience knows, the outermost layer of the heart is not myocytes, is this that looks like an epithelium, is this wrapping of cells that are protecting the myocytes from the friction with the pericardial fluid. And if you look at those cells, they look like an epithelium with these beautiful intercellular junctions, with beautiful desmosomes and gap junctions. So we thought, okay, we got it. The desmosomes are not working. The epicardial cells go into a transformative stage. They become fibroblasts, from fibroblasts to adipocytes, and there you go, ARVC. And we were able to convince NIH to give us an R01 to do this, and we then generated a mouse model with our same logs, so our same knockout, but to knock out plakophilin-2 only in the epicardial cells. And we took the mice and we made the mice. Eric Small in University of Rochester did this. It was a joint effort. And he did it and trained them and run them and spit on them, and they just don't have any phenotype. So you can remove the entire PKP2 of the desmosome of the epicardial cells and nothing happens. Now, interestingly though, if you put them in both the epicardium and the myocytes, then they do get a little worse a little bit sooner, but it's a very mild effect. So it looks as though in the mice, in this model, the plakophilin-2 in the myocytes play the major role and the epicardium less. But of course, again, that's just the model of the mouse.
Yeah, but in the case of Stefan, what he was talking about is that the epicardial cells actually had these progenitor cells that converted either into fibroblasts or in adipocytes, and adipocytes that later generated fibrosis.
Yeah.
So the epicardium does seem to have a role in atrial fibrillation.
Yeah. Now, we studied the epicardium in vitro, and that's the preliminary data of that grant, because in vitro, it works beautifully. If you take epicardium from a mouse and you silence plakophilin-2, in vitro, the proliferative stage of the epicardium, the transformative stage of the epicardium, is very affected by the loss of plakophilin-2. But in the mice, it doesn't have. When you put things into the system, and that is the importance of the system. As you know, I am cautiously optimistic, but very cautious about interpreting work in iPS cells, for example. Sure. Because in ARVC in particular, this is a multi-system disease, right? And lots of things are affecting it. So it's the same. In the dish, you can see things that then you put it in an adult mammalian heart, and it just doesn't happen.
You didn't see any phenotype in the mouse, even in the atria?
Nothing. And the atria, the epicardium, is far more active than in the ventricle. Right. For culturing it, if you put a piece of atria on a dish, the epicardial cells will migrate right away. If you put a piece of ventricle, it takes a long time. So it's much more dynamic in the atria.
Okay. Very good. Well, I think we have, unless any of the speakers has a comment or someone from the audience has a very important question.
Yep.
Yes.
A question here?
Yes.
Okay.
Sorry I cannot see you, but can you
Okay.
identify yourself?
Yes. My name is Gonzalo Pizarro. I'm from CENIC in Madrid. And
Gonzalo, very rapid question.
yes, well, thank you for the session, but my question is desmosomal reserve loss reversible, and is there any imaging or biomarker that could tell us here we are in an reversible state or not, maybe in the future?
Yeah, so the desmosomal reserve, if we have not done it, but I am going to assume that if we were to take these mice and give them 6 weeks or 2 or 12 weeks of rest, they probably would go back to the transcriptional program of the sedentary mouse. So in that model, yes. And indeed, patients with ARVC that stop exercising, my understanding is that the progression of the disease slows down. So there is an advantage to the stopping of exercise. That's I guess, the quick answer to a quick question.
Okay. Well, I want to thank the speakers for, this has been a wonderful session. I think we had a lot of variability in terms of inheritable cardiac diseases, and this has been very informative, at least for me. I've learned a lot, and I want to thank you all three for being here. And it's time to close the session. Connie, thank you very much. Mario, thank you. Juan, thank you very much to all three. And thank the audience. Bye-bye.